Hello everyone, welcome to Biolation. Today I will be starting a new topic which is yet another part of the chapter cell cycle and cell division. So today's topic is chromosome, its structure and function. So let's get started. So before we start this chapter, we need to recall few things that we have learned in cell cycle and cell division. So what happens in the interphase? The nucleus shows a network of very long, extremely thin, dark staining fibers, which are called the chromatin fibers. Now, as the cell begins to enter the first stage of cell division, that is prophase, the chromatin fibers condense to form the chromosomes. And this is what a single chromosome looks like. So my first question is, what are chromosomes? So chromosomes are highly condensed chromatin fibers which are formed at the prophase stage of cell division. These are small, thick, rod-like, darkly stainable structures. Now, stainable structures means the structure which can take stain or color. The term chromosome is derived from two Greek words, chrom, which means colored, and soma, which means body. So, chromosome means colored bodies. The structure of chromosome can be best studied during the metaphase and anaphase stage of cell division. Now, in case of non-dividing cells, these are seen as a network of thread-like structures which are called the chromatin fibers. So my next question is, why chromosomes are important? So chromosomes are important because the chromosome play an important role in cell division. Chromosomes help in the transfer of genetic information from the parents to the offspring during reproduction, and the chromosome undergoes mutations and contribute towards the evolution of life. Now, what are mutations? Mutations are sudden changes that occur in the chromosomes. So this mutation can contribute towards evolution of life. Next, my third question is, who discovered chromosomes, or how the chromosomes were discovered? So the chromosomes were first discovered in 1882 by a German scientist named Walter Fleming. Fleming noticed this in the rapidly dividing cells of larvae of salamander. Salamander falls under amphibia. Now in the microscope, he saw minute threads that appeared to be dividing lengthwise, though the microscope was of old type. And Fleming called their division as mitosis. Depending upon the number of chromosomes, the cells are of two types, haploid and diploid. So what are haploid cells? A cell is said to be haploid when all the types of chromosomes found in it are present in a single set, and this is what a haploid cell look like, containing a single set of chromosomes. Now, what a diploid cell? A cell is said to be diploid when all the types of chromosomes found in it are present in double set. And this is what a diploid cell look like, containing double set of chromosomes. Next, we will study in detail about the structure of chromosome. What are chromatids? Each metaphase chromosome appear to be longitudinally divided into two identical parts, and each of the identical parts are called the chromatid. Now, chromatids of a chromosome appear to be joined together at a point, which is called the centomere. The two chromatids which make up a chromosome are called the sister chromatids. So these two chromatids are the sister chromatids. And the chromatids of the homologous chromosomes are known as non-sister chromatids. Now what the chromatid is made up of? The chromatid is composed of two substances, DNA and histones. DNA are deoxyribonucleic acid. These are present about 40% in the chromatid, and histones are a particular type of proteins which are present about 60% in the chromatid. What are DNA? DNA is a very large molecule, therefore it is called macromolecule. Macro means large. Now DNA is composed of two complementary strands which are coiled around a common axis to form a double helix. Now each single strand is composed of repeating nucleotides, which are composed of three parts: phosphate, a pentose sugar, and a nitrogenous base. Thus, the two strands together make a ladder-like arrangement with the nitrogenous bases. The DNA molecules contain 4 nitrogenous bases, and these 4 nitrogenous bases are adenine, guanine, cytosine, and thymine. Now, adenine is denoted as A, guanine as the capital G, cytosine as capital C, and thymine as capital T. Next, how the chromosomes are formed from the DNA. The DNA strands wound around the core of 8 histone molecules. Now, if you imagine the core as a football, and around that football, a long rope is wound around with 1 or 2 loops. Now, each such complex, which is formed with the 8 histone octamer and the DNA wrapped around, are called nucleosomes. Next, many nucleosomes fold up to form chromatin fiber. The chromatin fiber again forms loops. Next, the chromatin fibers further compress, fold, and coil tightly into the chromatid of a chromosome. So this is how the DNA gets converted to chromosome due to coiling and supercoiling. Next, in each chromatid is present a highly coiled thread of DNA and proteins which are called chromonema. The chromonema bears darkly stainable granules which are called the chromomere. The chromonema is surrounded by matrix, and the matrix is covered by another covering which is called the pellicle. And the terminal regions on either side of the chromosomes are called the telomeres. Next, depending on the position of the centromeres, the chromosomes can be of 4 types: metacentric, submetacentric, acrocentric, and telocentric. Now, in metacentric chromosome, the centromere is located exactly at the center of the chromosomes. Now, these chromosomes assume V-shaped during anaphase. In case of submetacentric chromosome, the centromere is not exactly at the center, but it is near to the center of the chromosome. And during anaphase, these chromosomes become J or L shaped. Next, in case of acrocentric chromosomes, the centromere is located very near to one of the ends of the chromosomes. Thus, the chromosome acquire a J shape or rod shaped during anaphase. And finally, in case of telocentric chromosomes, the centromere is located at the tip of the chromosome. As a result, only one arm forms. Now, these chromosomes look eye-shaped or rod-shaped during anaphase. Next, what are the differences between the chromosomes and chromatin? Now, in chromosomes, the DNA are tightly packed. On the other hand, in chromatin, the DNA are not tightly packed. The chromosomes are found only during cell division, whereas the chromatin are found throughout the interphase stage. In chromosomes, the DNA is not being used for macromolecule synthesis, whereas in chromatin, the DNA is being used for the macromolecule synthesis. Autosomes and sex chromosomes. So what are autosomes and sex chromosomes? The chromosomes which determine the sex, that is the maleness or femaleness of an individual in asexually reproducing organisms, are called sex chromosomes. Now, sex chromosome has another two terms which are called allosomes or idiosomes. And all the chromosomes other than the sex chromosomes which are found in the pairs of homologous chromosomes in diploid cells are known as autosomes. So autosomes are found in both males and females. Now we humans have 23 pairs of chromosomes. Now out of this 23 pairs of chromosomes, 22 pairs are autosomes. Sex chromosomes determine an individual's gender, whether that individual will be a male or a female. Now 22 pairs are autosomes and the rest one pair is the sex chromosome, which contains the X and Y chromosomes. The X and Y chromosomes are not homologous. This is because the X chromosome is much larger than the Y chromosome and contains many genes. And the Y chromosome has only a small number of genes. In humans and other mammals, the females are X and males are XY. So here the Y is the male determining factor. So Y determines that the individual will be a male.